Welcome to This Explainer. Today, we're going to explore the absolutely fascinating, invisible, and honestly mind-bogglingly complex chemical communication systems that are hard at work inside your head every single second of the day. I mean, think about the sheer stakes of this for a moment. How exactly do billions of individual, completely isolated cells talk to each other to create every single thought, feeling, memory, and movement you'll ever experience? Right now, literally as you're comprehending the words I'm saying, a massive microscopic chemical storm is raging in your brain. Today, we're going to decode exactly how that storm works.
Okay, let's dive into this. Here is our roadmap for today. First, we'll look at the brain's chemical messengers. 2, the 7 steps of action. 3, fast versus slow receptors. And finally, 4, the brain's chemical symphony.
Starting with part 1, the brain's chemical messengers. So, there are 3 primary categories of these chemical couriers, and they're basically defined by how far they travel and how they deliver their message. First up, we have our classic neurotransmitters. These operate via what's called wiring transmission. They cross tiny, microscopic gaps between adjacent cells, so highly local, highly specific. Then we have neuromodulators. Now, these operate via volume transmission. Instead of just targeting one neighbor, they diffuse over entire regions of the brain to alter global states, things like attention or vigilance. And finally, we have neurohormones, which are released right into the bloodstream to travel vast distances to influence target organs far, far away.
The physical differences between these are pretty striking when you think about it. A classic neurotransmitter works across a private, highly localized channel, literally just 2 cells whispering to each other across a tiny synaptic gap. But a neuromodulator, it doesn't just whisper to one cell. It diffuses away from the site of release, interacting with multiple cells at a distance. Get this, neuromodulators can actually remain in your spinal cord for weeks, continually adjusting your brain's sensitivity and metabolic processing.
And this brilliantly illustrates the difference. Think of a neurotransmitter kind of like a private telephone signal. It conveys a very specific message from one sender to one intended receiver. Neurohormones, on the other hand, function way more like a radio station. They broadcast a general message out into the bloodstream, and any cell with the right receptor, basically anyone tuned to the right station, will pick up the signal. Moving on to part 2, 7 steps of action. Let's head into the synaptic gap. According to a framework outlined by biopsychologist John Pinnell, neurotransmitter action happens in 7 distinct steps. Let's track the birth and deployment of a molecule here. Step 1 is synthesis. The neurotransmitter is manufactured from precursors, often right there in the axon terminal. Step 2, storage. It gets safely packed away into these tiny spherical packages called vesicles. Step 3, breakdown of leaks. The brain is incredibly efficient. If any transmitter leaks out of its vesicle prematurely, enzymes quickly destroy it. And then, step 4, exocytosis. An electrical action potential hits, causing the vesicles to fuse with the cell membrane, explosively releasing their chemical payloads right out into the synaptic gap. Let's move along and see how this builds into the final stages. The chemical is now out there in the gap. Step 5 is absolutely crucial: inhibitory feedback. The releasing cell has autoreceptors that act sort of like a thermostat, monitoring how much chemical is in the gap and telling the cell to stop releasing if there's enough. Step 6 is the main event: activation. The neurotransmitter binds to the receptors on the receiving cell, delivering the message. And finally, step 7, deactivation. The stage literally must be cleared so the next message can come through. Let's look a little closer at that final step, deactivation, because it's genuinely fascinating. You can't just leave chemical messengers lingering in the gap, or the receiving cell would just fire endlessly. So synapses have 2 main methods for clearing the stage. The first is reuptake. This is a brilliant recycling program where special transporter proteins basically vacuum the intact neurotransmitter right back into the releasing cell so it can be used again. Now, if that doesn't happen, the synapse relies on enzymatic degradation, where enzymes ruthlessly chop up and destroy the transmitter molecules. Either way, the gap is neatly cleared. Part 3, fast versus slow receptors. Okay, so receptors basically come in 2 major flavors, and they completely change how the brain operates. On one side, we have ionotropic receptors. These are incredibly fast, operating in fractions of a millisecond, but their effects are very brief. They act as direct gates for ions. On the other side, we have metabotropic receptors. These are much slower to react, sometimes taking hundreds of milliseconds or longer, but their effects are incredibly enduring. Instead of opening a gate directly, they rely on complex internal messengers called G proteins. To really grasp how fast ionotropic receptors are, the researcher James Calat offers this great visual. Imagine a twisted paper bag. The millisecond the neurotransmitter binds to the receptor, it instantly untwists the bag. Boom, the channel opens and ions immediately rush in or out of the cell. It's direct, it's instant, and it's an immediate electrical shift.
Now, what's really interesting about this process is how we contrast that with metatropic receptors. Because they don't open channels directly, they're way more complex. Imagine you're outside a room, and you push a stick through the wall to unlatch a cage. An angry dog bursts out, running around the room, waking up a bunch of sleeping rabbits, which then scurry around, causing absolute chaos. That is a metatropic receptor. The neurotransmitter is the stick. It releases a G protein, the angry dog, which then travels through the cell, activating second messengers, the rabbits, changing gene expression, opening other channels, and fundamentally altering the cell's internal state.
Because of these mechanical differences, these receptors control completely different aspects of your human experience. Just think about it for a second. For vision and hearing, your brain needs rapid, up to the millisecond information. You need to instantly process a car swerving towards you, or the exact notes of a song. That relies entirely on lightning-fast ionotropic synapses. But for more enduring states, like the lingering taste of a really good meal, the enduring sensation of pain, or your deepest human emotions, arousal and pleasure, exact millisecond timing doesn't really matter. These fundamental states rely on the slow, lasting, widespread internal changes driven by those metatropic receptors.
And finally, part 4, the brain's chemical symphony. So the crucial point is this. Our brain is absolutely not a static computer board. As researcher Norman Deutsch highlights, neuromodulators, those slow-acting, widespread volume transmitters we talked about, do far more than just send a temporary signal. When chemicals like oxytocin or dopamine are released, they actually allow existing neuronal connections to literally melt away. They change gene expression. They allow for massive, large-scale reorganization of the brain's physical structure. Your chemical symphony is constantly remodeling the concert hall itself. Which leaves us with a truly fascinating reality to ponder. Every time you learn something new, every single time you experience a deep emotion, your chemical messengers are acting on those slow metatropic receptors to physically melt and rewire the architecture of your brain. So, if your thoughts are made of chemicals, and those chemicals are constantly changing the physical structure of how you think, who is really in control of your mind? Think about that the next time you feel a sudden rush of emotion.